All right, so this chapter is going to go into a little more detail about genetic crosses. In the last chapter, we talked about Mendel and the basic cross that you can do as far as just a regular big B, little b, and some different forms of that, codominance and incomplete dominance and all of that. All right, so Mendel didn't know anything about chromosomes, genes. He was basing everything he did on phenotypes, on the outcome, what they look like. So he said that there were these factors that organisms had, and then even though they had 2 copies of a factor, they passed on one of those 2, and so the offspring ended up getting a copy from each parent. Today, the chromosome theory of inheritance basically just says those factors, we're going to call them genes. And a gene is a specific location or a loci on a chromosome. And that, when he talked about the principle of segregation and the principle of independent assortment, that the factors or the units he was talking about, we're going to say that those units are actually the chromosomes that undergo segregation and independent assortment. So this is nothing new. It's just using the vocabulary that we know today and applying it to what Mendel said, back in the 1890 or whatever. All right, and you already know this also. In every what's a somatic cell?
Body cell.
Good, a body cell. So in every body cell, we have 23 pairs of chromosomes. 22 pair are going to be called autosomes. And what's the last pair?
The one with gender.
Right, the ones that determine gender, and those are your sex chromosomes, and we use X and Y to represent them. Now, keep in mind, and we're going to talk about this now, that the sex chromosomes, particularly the X chromosome, doesn't just determine sex. X chromosome doesn't just make you a female if you have 2 copies of it. In fact, it's more like a child would automatically develop female if they didn't have a Y. The Y changes that so that you become a male.
But like on those sex cells that determine gender, like are there traits on them, or is it just specifically for gender?
Well, that's what I was going to say. The Y mainly just determines gender. It's very small, and it makes you a male. The X has a whole bunch of stuff on it. You have to have at least one X to survive. That's why males still have an X. For example, in the last chapter we talked about color blindness. So that's on the X. So that means your color vision has to do with the X chromosome.
Okay.
Another one is hemophilia. Certain blood clotting factors are on the X chromosome, and if there's a mistake in that area, you end up with hemophilia. A third one is muscular dystrophy. Certain things about muscle development are on the X chromosome, and if there's a mistake there, you end up with muscular dystrophy. So the X chromosome has a lot of information on it that has nothing to do with your sex.
What causes a mistake?
Mutations cause the mistake, although in other words, some of these are recessive genes. How did it originally come about? Probably a mistake somewhere along the way. Today we just call it the gene for muscular dystrophy or the gene for hemophilia, but probably it originated as some kind of a mutation. Okay, so a gene is really just a spot on a chromosome. On the worksheet, they're simplifying it for you and having you count the bands. They're representing genes by bands. Actually, a band would contain many, many, many genes. If you've gotten that far on the worksheet, you see that certain chromosomes have thousands of genes. All right, so Thomas Hunt Morgan, you won't be tested on his name. He is the one who basically discovered sex-linked traits that were carried on the X chromosome. And he did this working with the fruit fly, Drosophila. Just like pea plants, fruit flies are very easy to work with. They have a really short lifespan, and they have traits that are really easy to see. As a matter of fact, they only have 4 pair of chromosomes. So I used to have people breed fruit flies. We would actually order them and cross red eyes and white eyes, and you could put them in a little vial. It's actually pretty easy, but they're very small, so they're kind of hard to work with. You had to use little paintbrushes to separate them. But if you allow them to mate and then let all the adults go, you'll see maggots. The maggots will make little cocoons. And then, if there's no flies in there at all, the day that they hatch, females don't mate for the first 13 hours. So if you came in and there were newly hatched flies, you would be sure that those were virgin females. So then you could take those females from that vial and then pick males from whatever you wanted to cross them with, and then you could cross them. So just like the genetics of the pea plants, they're really easy to work with. The reason we don't work with them anymore, or the reason I choose not to, some people still do, is if you look at the pictures, the TV has a better picture, The right side are males and the left side are females. And if you really look at them, there's not a whole lot of difference other than the males have a kind of a darker behind. The females have a kind of a more white behind. But imagine that's highly magnified. These are like gnats. So it's really a pain. Everybody get out their magnifying glasses and people would end up with like vials of all males and then they'd get no eggs and so it was a problem. Alright, so what did he do? So how did we discover it? Well, first of all, he would actually wait for a mutation to appear. I can order the flies. I can say, okay, I want a vial of white-eyed flies, or I want a vial of wingless flies, or whatever. The mutations are already there and I can order them. What he did is he just let them cross for many generations and then sometimes one would appear with a mutation. And so he called that a mutant, and he designated it with a W, and the rest of them, the normal ones, he called wild type, and he designated them with a W with a little plus. So once he created a mutant, he would just basically take the mutant and he would cross it with other flies and then just follow the results. Okay, a mutant came out with white eyes. I'm going to take that white-eyed fly and let it mate with these red-eyed ones, or let it mate with other, if it was a female, let it mate with red-eyed males, etc. And then he would just follow the results. So from that, here's an example of what he did. He crossed a white-eyed male with a red-eyed female. In the F1 generation, all the flies had red eyes, which tells us what? Good. Red is dominant over white, which is what he expected. There was not any big surprise there. This is just like one of Mendel's crosses. You could say, okay, so the male must have been little r, the female must have been big R. Everybody comes out big R, little r. The difference came in the second generation. In the second generation, all the females had red eyes, but the males came out 50% of them with red eyes, 50% of them with white eyes. So he knew this was not a normal cross because it was affecting the outcome on males differently than females. And so his conclusion was, this is a gene that's carried on the X chromosome. So when you're doing one of these crosses, and I gave you a quick walkthrough when we were working on the last lab, when you're doing one of these crosses, you have to write it so that the trait is actually on the X. Sometimes they're called X-linked crosses because there's almost no genes on the Y other than what makes a male a male. So there's it would be very, very rare that a male, because it would only be in males, would even have a disorder that was associated with the Y chromosome. So we usually call them X-linked traits because pretty much they're on the X.
Why do you think that it was on the X chromosome if it was affecting males? Wouldn't it be on the Y chromosome?
Actually, I'll show you. I'll show you. So this is, he worked it out mathematically, but I'll show you what happens here. So a female would be X with a letter, and then a male would be X with a letter Y. And then N would represent whatever the allele was, big B, little b, etc. So I was just using N for just a random allele. So if we go back to the cross that he did, I'm going to go back to his original cross. So give me a second here. Okay. Where is it? Here. Okay. So he would have said that the white-eyed male would be X, let's say little r, Y. In other words, he has an X, he has a Y, and he has a little r. Our female is always XX, and she's going to be X big R, X big R. We're going to assume because the mutations are rare. When we cross this, X big R, X big R, X little r, Y, you could still cut out one of those X's if you wanted to. With sex-linked crosses, I don't usually do that. Here's our F1 generation. So we get X big R, X little r, X big R, X little r, X big R, Y, X big R, Y. So basically everybody comes out with red eyes. Does that make sense so far? Now he crosses two of the F1s with each other. So all of our males came out X big R, Y, and all of our females came out X big R, X little r. So now if we cross these males with these females, look what we get. So our females come out X big R, X big R, X big R, X little r. They all come out with red eyes. But our males, X big R, Y, and actually I have the answer to your question. If it was on the Y chromosome, then every male would get whatever it was. And he didn't get that. He got 50% of the males with red and 50% with white. So if it had been on the Y chromosome, every male would have had it. And that's not what he saw. He saw all females with red eyes. And then half of the males red and half of the males white. Now, what if instead of that, he actually did a different cross? What if it was a female in his mutant generation? So what if he instead had a female with the white eyes and crossed it with a male with red eyes? What would be the genotype for the female with white eyes? X little r, X little r. And we're going to say that she's crossed with a normal male. Watch what's going to happen this time. So we get X big R, X little r, X big R, X little r. Do any of our females have white eyes?
No.
No. How about our males?
Yeah.
All the males, right? So all of our males would come out with white eyes, and all of our females would be normal. So who is the male getting white eyes from? In every single cross here.
The female.
He always gets it from his mom because he gets a Y from his dad. So if your dad had hemophilia or your dad was colorblind, and you are a boy, you would not carry that trait because dads cannot pass it to their sons. In order to be a son, you got the Y. But if he has it, if we go back to this cross, the dad has white eyes. Notice he passes it to all of his daughters. Now they're only heterozygous for it. So tell me this. How in the world would you get a white-eyed female? What would you have to cross to get a white-eyed female? At the bare minimum.
She would have to be his mom and
Good.
fully white.
Exactly. She would have to be a carrier, and dad would have to have it. Now, remember, most of these traits we're talking about are rare. Hemophilia, color blindness not as rare as some of the others. Muscular dystrophy, most of the traits that are carried on the X chromosome are extremely rare. So the chances of getting a female with it, having a male that has it, marrying a female, male that has it, marrying a female that's a carrier of it, extremely rare. Which is why you're going to see very few, if any, girls with an X-linked trait. They're much more common in males.
So what if the male did have it?
If the male doesn't have it, then there's no chance at all of a daughter having it.
Okay.
Well, that's not true. No, that is true.
Okay.
Even if the mom has it, the daughters are not going to have it. They're going to be carriers, right? Because they're going to get a good copy from their dad no matter what. But if the mom has it, all her sons are
But
going
let's
to get
say
it.
she was little r, and he was big R. What would happen?
If she's little r, that's this one.
And he's big R.
And he's big R. So none of the daughters can have it because they're all going to get a good X from their dad.
Okay.
But the sons are only going to get the Y from their dad, so every son is going to have it because they're going to get the bad X from their mom. So sex-linked traits are passed from mother to son. Going back over here. And, this is basically the cross. If we crossed a normal male here with a hemophiliac female, this is the cross that you would get. So notice again, our females, none of them would have it, but half of our males. And I'll go back to this slide if you're copying, because I think that's the cross that's in your notes. So if you're trying to write this cross down, I'll come back to this slide last. So just hold off. I just want to show you this really quick. Hemophilia in the royal family. Remember, in royal families, you were supposed to marry people that also had royal blood. So you see a lot of, in a pedigree, you see your males and females. You'll see, like for example here, these two are cousins. So a trait that's normally very rare, we actually see this trait a lot in the royal family. Now, do you see any females that have hemophilia in the picture? And to have it, you're colored in. If you're a carrier, you're half colored in. So do any females have hemophilia?
Victoria.
She's a carrier,
No.
right?
You have to be complete.
No, right, you'd have to be completely colored in. So again, sex-linked traits, more common in males, always given to males by their mother, and then we're gonna do one more practice.
So it must be like really, really, really rare for people to have it, because even in the royal family, like like they're most likely all carriers. So
Exactly,
they're
it's
gonna
almost
get each
impossible, and especially with hemophilia, because back then there wasn't a lot of treatment, so males that
Yeah.
had it probably died before they could pass it on
other. Yeah.
in a lot of cases. So you'd